My name is Will Dichtel. I am a professor here in the chemistry department at Northwestern. I can tell you a little bit about the kinds of work that we do, and then the talk will be about one specific effort within our group. So I'm an organic and a polymer chemist, and the overall theme of our research is to develop new ways of polymerization, new ways of organizing molecules through weaker interactions like non-covalent interactions, all to control the properties of materials and surfaces in ways that were not previously possible. And today I'm going to talk about our work in water purification, and it really started out as an effort to control porosity in materials that were in polymers that were derived from natural resources instead of petroleum. And so that is the talk today. So removing organic pollutants from water and using polymers derived, in this case, from corn. Okay, so what we are doing today is I'm going to be talking to you about these porous cyclodextrin polymers. And cyclodextrin is shown in kind of chemistry hieroglyphics over here. It looks like a big, somewhat fancy looking molecule, but if you look at little pieces of cyclodextrin, those are sugars, and in particular that sugar is glucose. So it's maybe the most common and recognizable sugar in biochemistry. And there are enzymes in nature and now that have been cultivated that will take cornstarch directly, literally out in the cornfields of Iowa. They make cornstarch, and then right next to that is the cyclodextrin factory. So they take cornstarch, which is a polymer of glucose, and they've got an enzyme that'll convert it into this compound beta cyclodextrin, which is a cyclic structure with 7 glucoses in it. And so that compound is in your everyday life, whether you know it or not. It's by virtue of it being this ring, the inside of the cyclodextrin is actually kind of oily and greasy, if you can sort of think about it in that way. And all of the OH groups of the sugar are pointing out into the water when it's dissolved in water. And so cyclodextrin, it's got this tiny little nano environment that is like this oily nano environment, but the overall molecule dissolves in water. And that pocket can bind a lot of the pollutants that we want to remove from water, a lot of organic molecules in particular. And it's already used in things like dryer sheets. It's used in the product Febreze. So if you don't like doing your laundry and you also don't like smelling bad, you can spray Febreze on your clothes and you will not smell as offensive. What's happening there is that these little pockets are binding the molecules that smell bad, and they're holding onto them tightly so that fewer of those molecules can reach your nose. And so, ironically, you're making your clothes dirtier by spraying them with Febreze, but you actually smell slightly better. And then their cyclodextrin is also capable of binding these pollutants out of water. But as I mentioned, cyclodextrin itself is soluble in water. So when you dissolve cyclodextrin in water, it'll bind these pollutants, but it won't remove them because the cyclodextrin will just stay dissolved. And so what we do to actually remove pollutants using cyclodextrin is that we need to polymerize the cyclodextrin into this network that then is insoluble. So I've got this kind of in cartoon form here where we've got these blue cyclodextrin cups, and we're going to link them together with other molecules into kind of this infinite structure that cannot be dissolved. But you still got those pockets that are empty so that water can flow through this material. The cyclodextrins can pull out the pollutants, and then the water that passes by will then be cleaner after that process. And that idea has actually been around for a while. That the idea of using cyclodextrin for water purification has been proposed for at least 20 years, if not maybe even a little bit longer than that. But as it turns out, the polymers that were made prior to our work really were not ever the best solution for removing pollutants from water, in that they did okay, but they didn't perform as well as other alternative technologies. And so what I'll show you today is that the key to taking these polymers and really getting the most out of them involved building porosity into these materials and the way that we did that, and really our big innovation in this space was to use rigid crosslinkers to generate pores. Now I'll talk a little bit about how we think about that in a few slides. But as a result, when we do that, now we get a cyclodextrin material that is really, really fast and has high capacity for these pollutants. I'm going to show you that by studying the fundamental chemistry of how these materials form, we learned something about the types of pollutants that they could remove. And moreover, we learned how to change the structure to tune the selectivity of the material. So there's certain pollutants that we specifically want to target. And now we have some insight into how to do that by virtue of changing the crosslinker, because the cyclodextrin can be modified, but it's, that's relatively expensive. Swapping out the crosslinker, you can go to a chemical catalog and have many different options for that. So that's a really nice way to do that. All right, so I'll show you our starting point here, and this was our very first entry into this. We're actually doing this work about 5 years ago and then published it about 4 years ago. In blue here, this is just a shorthand for cyclodextrin, and then this is the crosslinker here in red and green. It's called tetrafluoroterephthalonitrile. I will refer to it as TFN some of the time. And this is just a chemical reaction where an oxygen displaces a fluorine to form an oxygen-carbon bond. There are 21 different oxygens per cyclodextrin, and they just react somewhat randomly. So you get, many of the oxygens are unmodified in the final material, and then some of them are reacted with crosslinkers. And we call this P-CDP, which stands for porous cyclodextrin polymer. And it had a surface area of about 260 meters squared per gram. At the same time, we failed a lot at this too, but failure was kind of a good thing in certain ways. We were able to find conditions where we got slightly different forms of this polymer, including ones that were not particularly porous. So this one, NP-CDP, non-porous cyclodextrin, it has approximately the same chemical composition, but it was a low surface area version of this. So in comparing the performance of these two, that could give us some idea of the effect of the porosity on the performance of the material. The main difference between these polymers in terms of composition is that the porous structure is more heavily cross-linked. And so you can see that here in the table, the ratio of the crosslinker in these materials in the porous version is 6:1, and then so that's almost 1 per glucose. Remember, the cyclodextrin, each cyclodextrin has 7 glucoses, so it's almost 1 crosslinker per sugar in the porous derivative. It's 3.5:1 in the non-porous derivative. That's, basically every other sugar is modified. And that makes sense. The more you cross-link this material, the more rigid it is, and the more you sort of, the voids in the material that lead to the surface area can't, they can't rearrange. Like nature would like to pack down and fill all that empty space. But in this really gnarled up, highly cross-linked structure, it can't do that. So the material can't pack space efficiently. Okay, so a little bit about surface area. not everyone, in fact, many of you may not be exposed to these kinds of units or how we measure this, but one really good way to measure porosity in materials is by a technique called gas adsorption. So what we do is we take the material, we put it in a vessel with a really strong vacuum pump almost all the air out of there, and then we slowly dose in a certain gas and look to see how the pressure in the vessel responds to that. So in this case, we're using nitrogen at liquid nitrogen temperatures, and just as we're going through this this graph right here, as we're going to higher values of the x-axis, we're just dosing in more and more nitrogen. We're looking at if the nitrogen sticks to the surface of the material, it doesn't contribute as much to the pressure in the vessel. So a vessel with a really porous material in it will have a lower pressure for a given amount of nitrogen added. That gives you these traces once you actually build a machine that can do this precisely. And from that trace, you can, there are mathematical models that you can apply to derive this. So I mentioned roughly 250 meters squared per gram. There are 3 cyclodextrin polymers that we evaluated at first, the porous one, the non-porous one that we talked about, and then one that had been invented, as I mentioned, 20 some years ago, cross-linked by a different kind of more floppy linker, also a non-porous material. And in looking at how to do this in the context of, in water purification, what you think of like if you were to look inside your Brita filter or something like that, like a very common material to use is called activated carbon. And basically that's plant material that's been charred that also has a very high surface area. It's not a well-defined material, like you couldn't really draw a structure of it, but it's very high surface area. And we use 3 different grades of activated carbon to look at. These have different, different surface area values. The one in green labeled Norit is kind of a high-end, more expensive activated carbon, and it has a higher surface area. The red one is literally harvested out of a Brita filter. It's actually a combination of activated carbon and a few other materials in there. Doesn't, as you'll see, doesn't do a whole lot, and then there's sort of a middle of the road material as well. All right, so we looked at the ability of all of those materials to remove bisphenol A from water. So if you are drinking any liquid out of any container in the last 5 years, it's probably stamped BPA free. That's bisphenol A. It's thought to be an endocrine disruptor, and it's generally thought that you probably don't want to be exposed to it. And these 3 are the activated carbons, and you can see for this these chosen conditions, they bind some of the BPA very, very quickly, and then the rest of the BPA just kind of slowly adsorbs onto the material. You can apply another mathematical model to these data that where right now we're focused on the speed of that absorption. So there's this figure of merit, it's called K observed, which is basically a measure of how fast the adsorbent approaches its binding capacity. So higher numbers are better, and this says nothing about what the capacity actually is. It just tells you how fast it approaches whatever that capacity is. So I've added, so these were the activated carbons. You'll see the Brita filters down here, pretty low capacity. And then I've also added in green and in purple the non-porous cyclodextrin polymers, and you can kind of see why these never quite caught on 20 years ago, is that they actually look a lot like the activated carbons, but the activated carbons are way cheaper, right? It's way cheaper to burn plant matter than to synthesize anything rationally, right? So, the activated carbon is either coal or it's charred coconut shells or things like that. So that's pretty cheap. But if you stop and think about it, that actually is amazing that this non-porous material can compete on speed with these highly porous carbons. And that's because the cyclodextrin binding site itself is actually really, really good. It's just, its potential is not being unlocked because you've got these dense materials that are limited in their speed. Once you put a the porosity into the cyclodextrin polymer, it binds the BPA almost instantaneously. So it binds it in seconds, and it basically hits its capacity, in a minute or less. I mean, really, it's about 15 seconds where it's totally done. And so you can see for that figure of merit here, it's at least 10 times higher than these activated carbons, if not quite a bit more, 100 to 1,000 times in some cases. And so this was the first observation that we had back in 2015 that we were really onto something different and pretty special. And so we then said, all right, what's the coolest thing we could do with a material like that? So we turned our attention to a major societal problem. So this is a picture of the effluent of a wastewater treatment plant. So, remember, wastewater treatment, this is all the water that is after human use. So it's like you flush your toilet, or like your, water goes down the sink in your kitchen, or in many cases, like in Chicago, like just the runoff from our streets when it rains, it all goes to wastewater treatment plants. And we're very good at making that water safe in terms of removing or killing biological contaminants, right? Like you're not going to get cholera from this water after it's released into the environment. But this process was never designed to really remove trace organic molecules. And so it's been known for a while that all of the chemicals that we use to improve our lives, lots of different classes, they end up in our water, depending on exactly, what the water is used for and where. But these are things that, perfectly reasonable things for us to use, and things that are not going anywhere anytime soon, like pharmaceuticals, both prescription and non-prescription, flavors, fragrances, detergents, all kinds of industrial chemicals, pesticides, antibiotics, you name it. If humans use it, it ends up in our water, and these compounds leave our wastewater treatment plants at low concentration, but we then don't, in many cases, we don't even know what happens to them or what effects that they're having downstream. So this is a slightly more recent reference that just focused on pharmaceuticals in wastewater. These are various non-prescription and prescription medications, like this one, propranolol. A lot of people take it for high blood pressure. It was found, so this study tested the effluent of 50 wastewater treatment plants in America. So it's quite a pretty reasonable percentage of American wastewater. And they found it in 44 out of 50 effluents, and at this concentration of 260 nanograms per liter. So that is small, like really quite small. It's actually, this is a very potent compound in the human body. So, that actually is somewhat alarming. But then it's being diluted out into a larger body of water as it leaves the wastewater treatment plant. But we're starting to see that these compounds are, they're starting to make their way up the food chain. And like, every now and then you'll run into a study like they've tested salmon in the Pacific Northwest, and not only do they have these compounds in them, as they filter up the food chain, they've got like cocaine and caffeine and Splenda, and all these things are showing up in our wildlife. The other thing that is really alarming here is that in times of drought, we are very, very tempted to use this water coming out of a wastewater treatment plant and reuse it. And so, for example, in Israel right now, they're actually doing this as a routine practice. They're taking wastewater and using it for agriculture. And so essentially you're taking a complex cocktail of these, and these compounds are ending up in their agricultural goods, and then they're being directly eaten by humans. In other places, like, when California was in more serious drought than it is today, a few years ago, and will be again you know, and probably a few years down the line. Same idea is that there's temptation to use this water for agriculture or to take it to a drinking water treatment plant, which is also not really designed to remove these things. So we think it's important to develop ways to remove these trace organic compounds. And so we expanded our scale of what compounds we're going to look at with our material. So we teamed up with this environmental engineer, actually at Cornell University, named Damien Helbling. And he and his research group are experts on studying organic pollutants in the environment and in waterways. And they started studying the removal of these compounds down at the trace environmentally relevant concentrations that would be important if you wanted to apply this in a wastewater treatment plant. So this is a subset of 83 compounds that they studied in this first round with us. And they're just kind of representative of, lots of different, some of these are medicines, some of these are pesticides, some of these are flame retardants and things like that, sunscreens, and so there's a lot of data on this slide. And what I'll basically tell you is that first look at the left plot, so the one labeled pure water. And what we're doing is that we're comparing the porous cyclodextrin polymer performance against coconut shell activated carbon, CCAC. And the way to read these plots, you'll see several of these throughout the talk, is that when we're plotting a removal on an axis like this, it's one material versus the other. So if you're above the diagonal, our material performed better. If you're below the diagonal, the activated carbon performed better. And so you see in pure water that it's kind of a mixed bag. There are many of these compounds that are better on the cyclodextrin polymer. There are many of, there are quite a few that are better on the activated carbon. And it's, like what we notice is that there are a lot of compounds that are really good on our material, a lot of things up at 80 to 100%, whereas the activated carbon, it's more that it's kind of okay for everything, but not usually awesome at as many compounds. But then over here on the right-hand plot, it's the exact same experiment. It's just now the water is slightly more realistic, because instead of using like ultra pure water out of the lab, we started adding other things that are also going to be in the water in a wastewater treatment plant or in any sort of water that's exposed to air in the environment. So these are humic acids. So that's just kind of naturally occurring organic matter. Those stick to activated carbon as well, but they're very, very big molecules and they don't fit into the cyclodextrin cups. And so what we're seeing is that these are all the same pollutants, but when you've got other things in the water, that fouls the activated carbon and makes it perform much worse than it does under these pristine laboratory conditions. And so essentially what the biggest change in the right plot to the left plot is that the activated carbon stuff all shifted to the left with respect to the x-axis, and our material's better at removing almost everything. Now, the other thing on this plot that really, really caught our attention is that you see how some of these dots are color-coded. They're color-coded based on the charge of the pollutant. So the red dots are cationic. They're positively charged pollutants at this pH. The blue dots are negatively charged pollutants. The gray dots are neutral. And what you see in our material is that it loves positively charged pollutants. Like these are the best things here are positively charged, and the negatively charged ones are either some of them are okay, but a lot of them are really bad. And that was totally unexpected. We did not. We looked at that and said, there is something going on here, because when I was telling you about cyclodextrin a couple of slides ago, there was nothing about it being charged. Like the cyclodextrin is not inherently charged. I said it had this greasy environment, right? Like it should be binding other greasy molecules in kind of this like dissolves like kind of thing. So we said, okay, something's going on. And so we got to the bottom of this in two different ways. One way was a kind of big data approach, and another way was a very fundamental chemistry approach. So I'll talk about the big data stuff first. And we use this, this approach that is kind of borrowed from the pharmaceutical industry. It's called QSAR. That means quantitative structure activity relationship. It's a fancy way of saying we want to figure out, okay, like what attributes of a chemical structure make it likely to be removed well by our polymer, and also what attributes of a molecule make it more likely to be taken up well by activated carbon. Because activated carbon, you don't even really know the structure. So like this approach where you don't have to assume anything about it is really ideal. So I'll kind of quickly talk about how you approach this. The technical details are, really beyond what we have time for today, but basically it involved collecting a lot of data. So we studied 200 pollutants and their performance on our material and activated carbon. So we had a big data set. And because the pharmaceutical industry cares about this, they have figured out a million different ways to parameterize the features of a molecule. And so every substance out of these 200 has more than 3,500 descriptors. And so a descriptor is a quantitative, it's some sort of metric assigned to a structure that is just empirically chosen, right? Some of the ones that might make sense to us include the molecular size. Like there are many descriptors in there that deal only with size, don't care anything else about the molecule, like what's the volume, what's the longest length, what's the shortest length, all those things are in there. And then, what's the charge? How many hydrogen bonds can something have? Like what are all the atoms that it contains? Like people have found really brilliant and ways to put numbers to those so that you can apply machine learning models to them. So each molecule, 3,000 descriptors. All right, then we take all of our binding data and all of those descriptors and we feed it into this machine learning model. And the model figures out which descriptors contribute the most to the performance to removal performance. So out of those 3,000 descriptors, and so, sorry to get ahead of myself. So we take some of the data and we use it to train the model, and then we hold some of the data back, and then we use the data that we held back is called the validation set. And so then we apply that and we say, okay, like once we have a model, how good is it against the validation set? And fortunately, it's pretty good. This is activated carbon. So our model took those 3,500 descriptors and said only 14 of them are actually important for predicting the performance of activated carbon. And pretty good. And again, out of those 3,500 descriptors, only 8 of them the performance of our porous cyclodextrin polymer quite well. So so that gives us, so then we go and look at, okay, which 8 descriptors really matter. And so first for carbon, I said there were 14. One for electrostatic interaction, so positive and negative charges attracting. And, it's thought that activated carbon has some charges on it that we don't really understand, but that's not surprising. 3 descriptors for hydrophobic interactions. That is completely expected because that's what activated carbon is. It's this big greasy surface that, oily stuff gloms onto. So, and then size and shape, that also is not super surprising in the sense that they're tiny little pores in activated carbon. If you can't fit into the pore, you won't be able to access as much of the material. And then there's some descriptors that are hard to interpret physically. And so we don't get much insight into them, but basically just some constitutional, like the conductivity of the molecule. We don't really know what to make of it, but they do seem important. So no surprise, although this is the first time anyone's developed a model like this for activated carbon, it actually could be quite useful. For our porous polymer, these were, so five of the 9, 3 of them were electrostatic, positive and negative charges attracting. And that was really surprising because there were no charges intentionally put into that polymer by design. One descriptor for hydrophobic interactions, that makes sense, you know, like the inside of the cyclodextrin should, you know, is hydrophobic. And then size, that makes complete sense because the cyclodextrin cup is a very, very well-defined size. So if a molecule is too big, even if it would otherwise be a good match, can't fit. So, but the charge is there. Boy, I mean, that plus actually seeing that selectivity for cations really, really enlightening that something was going on. At the same time, we were studying this in our wet lab and saying, okay, you know, there are some things that didn't add up on our side either. So first off is that when we published this paper and we did this polymerization, the yield was very low. It was only 20 or 40%. And this style of polymerization should be very, very high yielding. Like the yield should be 80, 90%, 100% even, no problem. So that was weird. It was basically evidence that something else was going on. And also we were seeing that, you know, the polymer was clearly negatively charged when we studied that, you know, using what's called a zeta potential measurement. And then also another group made our polymer, you know, they used our exact procedure that we had published and noted that this material removed lead really well. And lead does not bind in cyclodextrin pockets like that. We didn't even test it because it would have made no sense to, but, you know, surprising. So, and lead is cationic in water too. So, okay, so from all that, we went and looked at this polymerization and we said, oh, never been reported before, but when we looked at this very carefully, we found this side reaction. And the side reaction is that, okay, this starting molecule, TFN, if you expose it exactly to our polymerization conditions, but don't give it any cyclodextrin, it forms this compound here, where there's one oxygen replaces a fluorine and it becomes negatively charged there. We say, aha, all right. That might be the source of why our polymer attracts all those cations, right? We never intended for it to be negatively charged, but it is accidentally. And furthermore, we went and studied this even more carefully by giving it an alcohol. So not cyclodextrin, but just butanol. And that kept everything soluble, doesn't make a polymer, but we can study the competition between that side reaction and the actual reaction that we're trying to do, which is forming these, you know, forming these productive bonds. And we actually found, you know, like this plot, you know, might be initially a little hard to follow, but basically this reaction is a competition. So, so it bifurcates several times. So this first reaction, you can make this side reaction product labeled 1B, or you can do the productive reaction to make 1A. The concentrations of those two species are in orange and blue, and we followed that as a function of time along this x-axis. So the productive pathway in blue, that product grows in, and then it keeps on reacting, so its concentration drains away. But the orange stuff, the side reaction, it forms, and once it forms, it just stays there the whole rest of the reaction. So it's dead. So when we said, okay, our yield was terrible, that's why, right? Is that we're losing a lot of our monomer here to the side reaction, and it's just getting washed out at the end of the polymerization. So we're throwing money down the drain. So then you start the blue pathway, and the blue pathway forms a second bond, and that's what you need in polymerization. You need at least 2 bonds to form, if not 3. And so that's good. That's in green. You see the green thing, green trace grow in after the blue one starting to drain away. But it can also make this red compound where you get this negative charge introduced here. That grows in. Again, once that happens, it's dead. But that is the way that you actually get the negative charges hooked onto the polymer structure itself. Like the orange thing, that stuff gets washed away. But the red one, if that happens with the cyclodextrin instead of in this sample reaction, that is going to be a negative charge that gets hooked onto the polymer and will never be removed. And that's how you get this weird selectivity. And then the green pathway can actually go forward and make a third bond, but that's fine. And in principle, you could even do the put a negative charge on that, although we never actually observed that happening. Okay, so having figured that out, we then figured out ways to modify the polymerization so that we can incorporate those negative charges in different amounts. So we could make a polymer that had a lot of the negative charges, and those are called phenols. So lots of negative charges. That's actually the bars here, is that by adding the base quickly or slowly, by changing the concentration, we could manipulate that. So we made 4 polymers, all the same composition, different amounts of negative charge in there. And then we correlated them to their ability to bind lead. And we saw that essentially it was directly correlated. So, and we also found this for organic cations as well. So this explained the selectivity. Now, here are lots of data. So now we've got low negative charges on the x-axis compared to a lot of negative charges on the y-axis. And this is a logarithmic plot. So going from 1 to 2 is a factor of 10 better performance. So, so this is really interesting. And you can see the cations are all here. They're these really high affinity structures, and they're bound better by the highly negatively charged polymer. But what's weird here is that everything binds better to the high negative charge polymer. Not well, right? Like all these blue anions are, this level of binding is not useful, but they're not below the diagonal. And so there's something else weird going on here with the ability of water to interact with these materials differently. It's not quite the perfect experiment, but it showed us something. Like we looked at this plot, and going into this experiment, we said, okay, if we, like once we thought we were just, we thought we had it made with this idea of like controlling the negative charge, because we said, okay, just dial it way down and we'll be able to bind anions. But this plot shows that that's not true. It's, the low phenylated polymer, we were hoping it would be, way over here, and we never saw that. So said, all right, if we want to bind negatively charged pollutants, we got to think of something else. And we really, really, really want to bind negatively charged pollutants. And here's why. There is this problem that is blowing up right now. When I started talking about this a few years ago, many people had not heard about this problem. Now there's a chance that you may have seen this in the news or seen it in the movies or things like that. So there's these substances collectively called PFAS. PFAS is necessary because this is kind of a mouthful. It's called per and polyfluorinated alkyl substances. So PFAS, they have been used in around the world for about 60 years now, and most notably in the manufacture of Teflon, in products that are waterproof and also oil-proof. So you think about that, like Gore-Tex, Scotchgard, things like that. That like if you're big into the outdoors and you buy all this gear that repels water, it's got this stuff in it, guaranteed. But also things like pizza boxes, like anything anything disposable that you need to put greasy food in is sprayed with derivatives of this stuff. So like the bowls at Chipotle that are paper and they brag are compostable are covered in this. Otherwise, you'd be really grossed out by your burrito bowl, like soaking through the paper as you ate it. So, sorry, bad image. Anyway, so one of the poster children for this class of compounds is called PFOA. There's a movie last fall called Dark Waters that starring Mark Ruffalo. It was basically like a Aaron Brockovich style thing where this lawyer took on DuPont for polluting this town in West Virginia. But this stuff we now know is toxic down at very, very low levels. It's not acutely toxic, but exposure over a lifetime is associated with cancer. It's associated with lots of complications in gestation and childbirth. It's associated with high cholesterol, and we are learning more all the time about these compounds. And there are thousands of them, but the ones that you'll hear about are called PFOA and PFOS. This is a map of last year of contamination in the United States. These things are environmentally persistent, like the carbon-fluorine bonds are very, very strong, and they're also not really found in biology. So there are not many or really any appreciable microorganisms that break these things down. So once they're in the environment, they're there for good. They move up the food chain. We've been putting them out in the world pretty indiscriminately for a lot of this time. 99% of Americans have detectable quantities in their blood. And you can see these dots are all communities that have water that are affected by these. And so Michigan looks terrible. Delaware looks terrible. Several other hotspots. The main sources of this are, so manufacturing. So like you can see Delaware looks like a pretty tough situation because that's DuPont, BASF, and major chemical companies there. Michigan is Dow, in Midland, but also lots of other industrial uses of this stuff. The purple dots are all military installations, and these things are used to put out jet fuel fires, and so they've been sprayed at thousands of Department of Defense sites for many years in training exercises and actual emergencies. So it's big. I mean, this is thought to be in the water at detectable concentrations of about 100 million Americans, so a third of the country or so. Okay, there's a lot of, I won't go over every one of these developments, but you'll now that I've pointed this out, if you have not noticed this in the news, you will start seeing this in the news. There's a lot of litigation on this. I mean, there's $1 billion and more in various judgments in places. The United States has really been woefully slow to regulate these things, and now states are actually taking action because our EPA is not really functioning appropriately right now to set more reasonable standards. But we basically need to remove this stuff down to nearly undetectable concentrations everywhere. Okay, so that's a long introduction to say, okay, we really want to take these polymers and remove those pollutants with it. And our first polymer did not, because a lot of these compounds are negatively charged. So our first polymer was no good for these anions. So that's kind of the structures on the bottom. Along the top, we've got things that are, basically what we did is that we changed the crosslinker. So this is a compound called decafluorobiphenyl. We can polymerize that just as well. And our hypothesis was that a more fluorinated polymer would work better. And we were right that more fluorinated polymer would work better. So this is for removing PFOA at environmentally relevant concentrations. The DFP, CDP, that's our new polymer. It removes it down to our detection limit. CCAC, so that's an activated carbon. So we're beating the best thing out there. And our first polymer doesn't remove it at all at these concentrations. So dramatic difference in performance. This is the EPA level, which is thought to be way, way, way too high. PPT is parts per trillion. So that's nanograms per liter. So like this is like a drop of PFOA in an Olympic sized swimming pool kind of levels that you need to get down below that. This is New Jersey is one of the states leading the way on appropriate regulation. That's their limit. And so, this material looked pretty good. One of the nice things about these is that you can regenerate them. So once they're full, you can wash them, you can wash the pollutants out. We use methanol or ethanol, and then just use it again. So, so we envision using these things repetitively. They don't lose any performance upon washing. But decafluorobiphenyl is a, like in terms of learning from it, we actually did learn something from it, but that is a compound, like the actual crosslinker is sold, you know, you can't buy it at an industrial scale, and to make it in, you know, the multi-kilogram quantities that we needed was not economically feasible, even for the severity of this problem. So we needed a new strategy, and that's where this charge idea came in. And if you're, you know, if you read up on our work at prior to this seminar, the other major thing, or another major thing that we do, are studying these two-dimensional polymers called covalent organic frameworks. And here what we did is that we actually studied them for removing these PFAS compounds. And the key was that we were putting, we're taking a very highly porous material and we're putting positive charges on it. So kind of in line with the theme of the rest of the talk, is that we thought that a material with greasy walls and a charge might interact well with these PFAS compounds. And that proved to be the case. So in this covalent organic framework, we had not just PFOA, but we tested 11 or 12 of these things, and they're removed in seconds, and they're removed quite well. Even these ones that are lower are actually the hardest ones to remove. So this performance was very, very interesting. And so we're very excited about it. But even those COFs are not really ready for making on huge scale yet. I mean, I think they will be someday, but they're not going to solve this problem on the time scale that is needed. And so we said, okay, we've got this cyclodextrin polymer. You know, what we haven't played with yet, there are these carbons triple bonded to nitrogens in these structures. And we realized that there's chemistry known to take those and make them into these functionalities called amines that will be positively charged in water. And so it was to say, oh, you know, in the original polymer, we accidentally put negative charges in it, but now we can rationally put positive charges into this material and see what happens. And so, you know, we're able to do that. We're able to prove that that those reactions were successful. And then we started looking at the selectivity. So this is a selectivity profile of the original polymer. You know, last time you saw it in a slightly different form, but it's essentially the same. It's, you know, awesome at cations, not good for most anions. And then these are the anionic PFAS compounds, and they're particularly pathetic at removing those. And then some neutrals that happen to fit cyclodextrin also come out well. After you do that reaction to the polymer, you change that functional group, the selectivity flips completely. And now, for some reason, there's one cation that binds really well. You know, we don't know why that is, but for the most part, the anions all become strong binders, and the PFAS especially become really, really well bound by this material. So, you know, really, really awesome to see that. So like, this is kind of the comparison plot that we were looking at before. You know, now we actually have something even better. We've got one polymerization, and then, you know, and then you take the product of that, and if you want the other selectivity, you do one more reaction to it and you completely change the selectivity. And the PFAS are here. Remember, this is a log plot. So like, being up in this corner is like really, that's where you want to be. All the anions on this side of the diagonal, the cations for the most part on this side of the diagonal. So these are well-balanced complementary materials. You could even use them as a physical mixture to separate everything. You know, if you want to remove both cations and anions, you don't need to develop a wonder material. You just use a mixture of these two, and you can get them both. Okay, so let me, you know, I mentioned a little bit about like actually trying to solve this problem, like for real, you know, not just like in the academic lab. Like, we want to go solve this problem. And so let me show you how that works, you know, especially as you're thinking about you know, maybe a career in science or like thinking about training at the graduate level, and, you know, want to give you the perspective of what basic research is and is not, right? So, so this is a cartoon picture of that cough, right? That was the thing that showed us, yes, this idea of putting nitrogen in your polymers would work well. Great to study, you know, lots of great fundamentals to it, too expensive to make. So we published that. But we learned something, and then we took that to our cyclodextrin polymers, and, you know, those are harder to study because the structure is not quite so well defined, but, you know, easy enough to, you know, we can do some understanding with them, but they're less expensive, and we could basically use what we learned and make a material and figure out that that works well. We published that one too. All right, in 2016, I started a company called Cyclopure, and Cyclopure is in the business of commercializing this discovery and actually putting it into technologies such that, you know, a consumer doesn't have to understand the chemistry. They just want to buy the equivalent of a Brita filter, or, you know, or we want to install this at the municipal level and, you know, have it be completely unknown to someone downstream of that their water is actually being purified in this way. So to do either of those things, you need to make a ton of this stuff. And there's some further design in here that, you know, like the materials that Cyclopure is using are not exactly what I've shown you, but they do use the same design criteria. There's some intellectual property, there's some patents that are involved in that. And so in response, they made a material that they now sell known as Dexorb Plus that derive from these discoveries. Now, not only did they have to make that material, they had to engineer it into a form that would be useful, right? And so there are a few different forms that would be useful. One form that we are using right now is that you actually incorporate this material into filter papers. So now we've got a paper that has this stuff woven into the paper. So you can just pass water through the paper and the pollutants are removed as the water passes through. From that, we launched our first product in November of 2019 called a water test kit. And so essentially what we were doing is that we were making a lot of this stuff, but still not enough to actually make consumer facing, you know, removal products. But we have made a test kit. So the way this works is that you take this filter paper in this kind of very highly engineered and designed device. It's a cup with the paper in it. You pour water through it. The water flows through in about 10 minutes. You mail it back to us. We can wash the pollutants off the thing. So then we wash the pollutants into our mass spectrometer and tell you exactly how much of these PFAS compounds that you have in your water. So we test for 35 of them. This thing costs $75. If you wanted to test your water normally, you'd have to find a not really consumer facing water testing lab and pay them about $500 to do that. And so very few people actually take the initiative to do that, but this is a very simple solution. That then allows us to scale this up even further and actually make removal products. So Cyclopure will launch a commercial removal product late this year or first quarter of 2021 that will contain this material and be drop-in replacement to popular consumer pitchers and under-counter things. So this is basically the evolution of like how you get from academic chemistry lab to like someone's house with this idea. And this is fast. Usually it takes 10 years or 15 years to go through this cycle, but the urgency of this problem and fortunately the material working very, very well and very, very talented team at Cyclopure have all made this happen faster than it normally does. All right, I will actually, I've got many other things to, kind of basic projects to mention here, but I'm going to actually stop. Just two other things here. Like I did mention Cyclopure, and that involves like, you know, big reactors and scaling up, and, you know, like we're making this, like when we first made this first material in my lab, we made 50 milligrams of it, and we struggled to give anyone a gram. Now we're making 60 kilograms a month of this stuff, and we'll be making even more of it later. I have to tell you that I will benefit financially if Cyclopure continues to follow that path. But I also want to talk about my research group and also the support for this work. This has been a project that we started in 2015. It was started by one postdoc in our group at first. His name is Aladin Alsbayi. He's now at, he now works for DuPont. He worked with a couple of other members of our team at the time. Brian Smith, who's not pictured here, and then several young students. So the students who first worked with Aladin have now just graduated. So Max Clemis is right here. He left our lab a month ago and took an industrial position at Intel. He did this work on PFAS stuff, like the Cyclodextrin PFAS work. Lele Zhao left a year ago and took an industrial position in a pharmaceutical company in Shanghai. Diego Alzate Sanchez was a student who was actually in Cornell's Department of Fiber Science collaborating with us. And then when we moved to Northwestern, it actually made sense for him to join our group and move with us. He did a lot of work on making this stuff practical and getting columns made of this thing so you can just flow water through them. He is now a postdoc at the University of Illinois at Urbana-Champaign. More recently, we've got current members of our group, Brittany Trang, who wasn't at our barbecue that day. Luke Scala has developed materials to remove chloroform from water, which I didn't talk about. And then Anna Yang is also working on PFAS. Finally, I mentioned Damien Helbling at Cornell. He's in environmental engineering. He started out with one graduate student on this project, Yuhan Ling. Yuhan has since graduated. He got his PhD at Cornell and he moved here to work for Cyclopure. So he's Cyclopure's head of engineering right now. And then this work was funded by the Center for Sustainable Polymers at the University of Minnesota. That's through the National Science Foundation. CERDIP is an effort by the Department of Defense to address PFAS contamination, and then also by the Dreyfus Foundation. So I'll stop there, and I'm happy to answer questions.